Thank you very much, Antonio. Thank you very much. Okay, let me just bring up my slides. Here we go. Okay, good afternoon, everyone in Busan, Korea. It's 2 a.m. here in Oklahoma, and I'm really excited that I'm here with you guys. And I basically need to thank four major figures that have been really kind of critical and important to kind of arrange all the things that we have for ISAM Neuroscience Interest Group meetings. And I think that I'm basically the last speaker of the last symposium of ISAM Neuroscience Interest Group. And I should thank those people. I mean, Dr. Song Gong Kim, who is the president of this meeting, he was really, really helpful for us to be able to arrange all these meetings in the same line, in the same hall. And I kind of significantly appreciate his supports from during last 6 months of arranging all these things together. And also Dr. Tai Yun Huang from National Center for Mental Health, who was really supportive for us to be able to be in contact with colleagues in Korea. Dr. Alex Baldacchino, as you know, he has been kind of really important in arranging all these things together. And then Antonio, who is a leading figure. I knew him for a long time as a leading figure in the field, but also he's a really great friend and supporting colleague. And I hope that we can kind of move forward together for Neuroscience Interest Group together. But what I'm going to discuss kind of in the next 10, kind of 15 minutes, I do not have any kind of disclosure of conflict of interest. We have been publishing in the field of brain stimulation and drug addiction, but I'm not going to discuss about what we have done in terms of how brain stimulation, how non-invasive brain stimulation might be helpful for the field of drug addiction. But what I'm going to discuss is give you another idea how kind of brain stimulation might be helpful for the field. But before starting the kind of the idea, I'm going to give you an idea about what are the publications that we already have in the field. And before you need to have kind of an idea about different types of brain stimulation, I know that Dr. Daughters discussed about kind of types of brain stimulation probably, but just to give you an idea, we have two major types. One type would use the electrical stimulation, and the other type would use magnetic stimulation. So we have transcranial electrical stimulation and transcranial magnetic stimulation. And for transcranial electrical stimulation, we mainly used direct current. So we use that at the term TDCS, transcranial direct current stimulation. And for TMS, in most of the time, we use repetitive TMS, so we use the term of RTMS. But in terms of the numbers of the studies, we have recently done an intensive systematic review finding studies that are people contribute to the field, and we already have 84 trials that people have done with TDCS and RTMS. Okay, so we already have 84, and there are 3 systematic reviews right now outside showing that there is a positive effect and hope for having a significant effect for TDCS and RTMS in the field of drug addiction medicine. But how different countries are contributing in these 84 publications? This is how we have contributions from different countries. We already have publications from 14 different countries in the field. It's interesting that you know that how much of the publications that we have in the field, I mean, how much of the scientific production that we have in the field is being contributed by US scientists to the field of drug addiction. If you kind of take a look inside the publications in the field of drug addiction, US contributes something like 45% of the entire publications in the field. But as you can see here in the field of brain stimulation, interestingly, we have something like 20% contribution from US side. And we have many other countries that are already active in the field. And we have kind of similar scenarios for other kind of innovative fields in the field of drug addiction. I'm going to discuss about them later on. But in terms of different technologies that different countries are using, you can see that TDCS studies in orange and TMS studies in blue. You can see different countries contribute to different types of kind of stimulation, but you have contributions from different countries with different stimulations. But in terms of different drugs that people are targeting in different countries, if you want to have a kind of idea about how different countries are contributing to the field, let's see, for example, what is the orange color? What is the drug that would represent the orange color? We have kind of drug orange from USA, from Italy, from Brazil. What is orange? Okay, so orange is cocaine, and then green is nicotine, and then dark blue is meth. So you can see that different countries, based on the major problems that they have, they are contributing, I mean, they are targeting different drugs for their studies. And in terms of the registered trials across the world, even if you just take a look inside the clinicaltrial.gov, you can see that people across the world are contributing to the field. And you know, clinicaltrial.gov is not the only kind of clinical trial registry, and there are many other clinical registries as well, like what we have with WHO. But even in the clinicaltrial.gov, which is the US-based registry, we have contributions from different countries. And then we have, in the field of even RTMS, we have contributions from countries across the world in the field. So we already have many countries actively contributing to the field. And in terms of using TMS or TDCS for different drugs, we have been, I mean, we are using both technologies for different types of drugs. So there is no specific preference for a specific type of drug. But right now, we are facing 5 major challenges to be able to really make a clinical impact in a way that we can use these technologies in the clinical practice. I'm going to give you an idea about these challenges and how we might be able to solve these challenges in the future. The challenge 1 is about the area to target. We are not still quite sure what area would be the best target for brain stimulation in the field of addiction medicine. And if I give you an idea about areas that people are targeting right now, most of the studies, I mean, the majority of the studies, they are targeting left DLPFC, left dorsolateral prefrontal cortex, with both TDCS and TMS, and they excite, I mean, do excitatory protocols. And in the second rank, there would be right DLPFC. But other than left DLPFC and right DLPFC, there are many other areas that people are targeting, and they're exploring potentials for potential hopes for drug addiction. We have published a paper discussing about different montages that we can have. For example, for electrical stimulation, we have many different options for targeting different areas inside the brain using electrical stimulation. And we have recently published a new trial showing that even kind of changing minor things in those montages in prefrontal cortex, we can have differential effects among drug users. So we are still not sure which one would be optimized and which one would be the best target for us. The second question is about dosage and how many sessions that we need to run. We need to get sure that if we want to have a significant effect for addiction medicine, we probably need to have 15 up to 20 sessions. But most of the studies in the field right now, they are just single session studies. They are basically experimental studies that are exploring potentials for having any effect. But in reality, if you want to do any clinical kind of effect, we need to have multiple sessions to be able to make a clinical impactful effect. Like any sort of other types of interventions like medication, one single session might not be that much effective. And then how long to follow up? Most of the studies in the field, they do not have any follow-up. So that is another challenge in the field. We really do not follow people in long term. And then the next question would be when to stimulate in the process of recovery. Should we start before detoxification? Should we start after? How long should we wait to start the stimulation? And studies that we already have, they targeted different stages of recovery. We have trials that targeted pre-treatment, those who are treatment seekers, those who are not treatment seekers. They targeted people during detoxification or in the later stages of recovery. So we are not quite sure which kind of time would be better. And even in terms of different drugs, we know, for example, almost all nicotine studies, they are targeting the time of kind of before starting the real treatment. So they hope that people who are treatment seekers or non-treatment seekers, if they receive a stimulation, they would have a higher chance to start the treatment and be in treatment. But they do not do a stimulation during treatment. So you can see there are differences between different drugs as well for that. And the challenge 4 is about how many subjects should we have in each arm of a clinical trial to have a meaningful results. There is a really important paper. If people are going to work in the field, I really highly recommend people to read the paper. But the bottom line of the paper is that if we have a sample size, something like less than 30 subjects in each group, we probably, if we report something, we would report something with larger effect size that is not really there. And if we kind of target one specific treatment outcome, there would be a high chance not to find the effect. So we need to have more than something like 30 subjects in each group to be able to have a power of something like 80 in the field. And if you take a look inside the studies that are published in the field, majority of them are less than 30 subjects in each arm. So it's really hard right now to really make a conclusion about, I mean, we already have 84 studies from different countries, but it's really hard to say, is there any effect there or not? So that is a major challenging issue in the field. And the last challenge is about the treatment outcome measure. What would be the reasonable treatment outcome measure in the field? And as you can imagine, many studies, they use drug craving as their target. But even for drug craving, people are using, even for just baseline drug craving measures, people are using something like 18 different types of questionnaires for drug craving. And there is no consistency in terms of which one would be better. And we know that most of them are highly correlated to each other, but we are not still there that which one would be an optimized kind of measure. And then people are doing drug relapse abstinence, that's their kind of outcome measure. And even for that, there is no consistency for that. And most of the studies are using just subjective self-reports for drug use. And the objective measures are really in minority. And then other outcome measures. People are using negative balance, positive balance, other outcome measures. And we still not quite sure which one would be the best outcome measures in the field. Okay, so these are 5 major challenges that we have. What do we think what are the solutions to have a better kind of contribution to the field? Because we have been working for something like 10 years and we have not been that much successful in making a really clinical impact. So the question is that, okay, how we might be able to not to work for another 10 years and coming back and see that we are still just confused about what are the things that we might be able to do in the field. So we realized that we need an international network of labs that are working in the field. So we started this idea of putting people together and talk to see how we might be able to collaborate across the world to have a harmonized system for a specific research matrix in the future to make an effect. So we started with a leadership core with kind of major figures in the field who have published kind of several times and are kind of key opinion leaders. And we have Stacy Daughters. I mean, I'm really kind of proud to have Stacy there, and she probably could give you an idea about what we are doing in the field. So we have started with this idea of putting people together, and then we received lots of really positive feedbacks from people across the board. So we already have around 50 labs across the world that are interested to collaborate from 19 different countries. And that's really amazing that how people are interested to collaborate and have shared protocols, shared ideas. We had a mixer meeting in the neurostimulation meeting in last August discussing about ideas, and we had meetings with people in NIDA, with NIMH, with NIAAA, how we might be able to have their support as well. And right now we are writing a consensus paper together discussing about things that we can do together, kind of specific clinical outcomes, what are the challenges in the field, and how we might be able to collaborate together to increase the quality standards and also targeting safety issues in the field. So we realize that it's something doable and we can arrange these sort of kind of collaborative network and put specific things outside. And even right now we are discussing about a specific checklist that could increase the quality of the publications and designing trials in the field. We are still discussing. There are many challenges when you want to have those types of checklists in the field, but there are kind of specific activities in making specific checklists in the field as well. So my last slide. What would be the future studies? I mean, what would be the ideal scenarios for future studies in the field? And the reality is that I think that what we are doing in the field of brain stimulation right now could be a model for other cognitive interventions. And I think that ISAM Neuroscience Interest Group should be a basic platform for these sort of international collaborations. So we hope that future studies should be reported in a way that would be 100% replicable. They should be pre-registered in a way that we know that what are the analyses that we are going to do. How we might be able to have online sync databases to collect data and lock the data without any potential to change the data in the next steps. How we can have shared outcome measures. How we can have shared data analysis protocols and platforms. How we might be able to have a shared matrix of research. How we can have multicentric trials, the trials that could be independent or kind of internationally funded. And we already have lots of great ideas how we might be able to run these types of kind of studies all together with collaborative partnerships. We have, as I mentioned in my talk in the morning, we have a special issue and frontiers that we are accepting kind of ideas and articles in the field. In the next brain stimulation meeting in Vancouver in February, we have a specific panel for international network of labs that are doing the brain stimulation for addiction medicine. So those who are in Canada, they are interested to be in the field, they can kind of join us. And I think that Neuroscience Interest Group in ISAM should be the basic platform for being able to run these sorts of studies. And I seriously feel that we can discuss about those things. We have a social meeting after, I think it would be on the fourth day of the Congress. And I hope that people can start a discussion on how we might be able to have other international collaborative networks in the field of kind of neuroscience for addiction medicine and how we might be able to collaborate together for that. And that's it. I really try to be on time, and I'm really happy to join you for questions.
Thank you very much. It's incredible that they have produced this presentation 2 in the morning. So thank you very much. Questions everyone to ask panelists? Okay. I think I don't have any particular question, but I think it was a fantastic ending to the series of symposia that we have had for the launch of the Neuroscience Interest Group. I found this presentation particularly interesting because it gives us a guideline of what we can achieve through the Neuroscience Interest Group. So I can only thank you again for being such an inspiring and driving force in this new community. And we hope to make more and more advances. So the network meeting is on Tuesday. 2:50 p.m. to 5:00 p.m. And it's in room 8. So we hope to see you all of you and some more people there. continue the discussions. Thanks a lot.
Thank you. Thank you. Have a nice day. Enjoy your time in Korea.
Well, we have to tell us where's that conference with the palm trees that you